Hi everyone, it's Jamie Batch, your instructor for AMP2. We just finished talking about how carbohydrates are digested by our digestive system and how they're absorbed through protein channels with the use of insulin hormone. We're now going to look at what happens to those carbohydrates, those simple carbohydrates slash monosaccharides, when they are absorbed into your cells. So I'm going to switch here. Here we go. That's where we left off. This is all going through the process of glycolysis. So glycolysis is an anaerobic process. Anaerobic refers to the fact that there is no oxygen being used. So oxygen does not have to be present. This can take place in an environment that's devoid of oxygen, or if your cells are having trouble absorbing oxygen or your heart's not pumping enough oxygen, etc. The overall process of glycolysis is breaking down glucose, which is a 6-carbon molecule, into 2 3-carbon molecules. So you would think, you just split the glucose molecule in half, right? Not so much. As everything in biology and chemistry is a little bit more complicated than just simply breaking the molecule in half. So there's a series of sub-steps similar to the citric acid cycle, where we had this kind of oxidation and reduction occurring along the way. Ultimately, there are 2 ATP molecules that are gained out of this. It actually uses 2 ATP molecules and generates 4 ATP molecules in the process, the whole process of glycolysis, so there's a net gain of just 2. It also releases 2 water molecules. So let's look at the overview here. So this is the whole thing. Do you have to know the whole thing? You don't have to know all of the sub-units, but you should understand the general process, the starting and end products. So let's start with kind of a big picture, all right? Maybe I'll stop screen sharing so you can pay attention to me instead of what's on the screen. So glycolysis as a whole starts with a couple main ingredients, and this is what I would do if I were you, and this is what I would do in a typical lecture. I would have the word glycolysis on the board. So write the word glycolysis somewhere around the middle of your paper. We're going to make like a little graphic here. So going into glycolysis. So these are words, and the arrows are going to point on to glycolysis. These are things like NAD. NAD goes into glycolysis. So you have the word NAD and the arrow pointing into the word glycolysis. What that's telling you is that NAD is needed in glycolysis. You also need the obvious, right? Glucose. Where does the glucose come? From your food, from your digestive tract, right? Glucose, arrow going in to glycolysis. So you have glucose and NAD. You also have inorganic phosphate and ADP coming in. So those are your 4 main ingredients. You also have, I'm looking at my graphic here in my little notebook. You also have some important enzymes that are used in glycolysis. One of them is called G3P. G3P. That's going into glycolysis. Another one is hexokinase, going into glycolysis. So you have inorganic phosphate, you have NAD, you have glucose, 2 enzymes, G3P and hexokinase. You also have 2 ATP going into glycolysis. So those are all of your, so to speak, reactants. What's leaving, what's being created, so these would be arrows coming off of the word glycolysis. You have 2 pyruvate molecules. That's your overall reaction, 2 pyruvate molecules. You also have 2 NADH molecules. You also have 4 ATP molecules. So coming out and leaving glycolysis, you have NADH, ATP, and pyruvate. Let me go back to sharing my screen with you so that you can see all this. And if you kind of look at this image and calculate, I guess one of the other things that you can add to that graphic is water. Water is another byproduct leaving glycolysis. We summarize all of our energy steps. Overall, we're inputting 2 ATP molecules. Notice the first few reactions are what we call endergonic reactions. They're ones that are using energy. ATP is actually being used up in the first few steps of glycolysis, in so much that if ATP is not present, obviously this the series of reactions will not occur. So you can, as I always suggest, print off each of these slides individually so that you can summarize them and really analyze them at a deeper level, explain what's going on, maybe even take that little white box off the side that says steps in glycolysis, cover that up, maybe fold the paper in half and see if you can summarize it on the one side of the paper, and then check your answers on the other side. So in general, you have, as soon as the glucose molecule comes in, the phosphate is attached to it. That process creates a molecule called glucose 6-phosphate. Glucose is basically phosphorylated, right? Popping a phosphate on to glucose is phosphorylating glucose. It uses ATP energy to do that. Another ATP is used when we phosphorylate it again and produce fructose 1,6-biphosphate. This molecule now is able to be split into glyceraldehyde 3-phosphate, into 2 of those, right? This is going to release hydrogen ions, and those hydrogen ions are captured on NAD to form NADH. Now our 1,3-biphosphoglycerate is going to be broken down again. It's going to be dephosphorylated or unphosphorylated. The phosphates are going to pop off. That is going to create ATP. The phosphates are going from that 1,3-biphosphoglycerate onto an ADP molecule to create ATP. The next step is to reduce 3-phosphoglycerate again to form water. And now we have phosphoenolpyruvate, which will then be unphosphorylated and create ATP and 2 pyruvate molecules. These 2 pyruvate molecules will go onto the mitochondria, where the citric acid cycle will take over, and that little sub-step will come on where a hydrogen is lost to form NADH, and coenzyme A comes on to form acetyl-CoA, and it's converted into a 2-carbon compound. So, summary of ATP production from glucose. In glycolysis, you're getting a net of 2 ATP. Overall 4, but you're using 2, so it's a net gain of 2. Electron transport system, citric acid cycle, gaining you around 34 ATP. 4 of them, again, from glycolysis, 24 from the citric acid cycle, and generated from the oxidation of NADH, 4 from the oxidation of FADH2, and 2 from GTP during citric acid cycle. So total, we're getting 34 ATP. Again, that number is an estimate, and new estimates say that's actually around 30. When I learned this back in my day, it was actually 36 to 38 ATP, and they keep lowering it. So I don't know. Either way, here's the whole process in a nutshell. Inside of a cell, you see glucose, a 6-carbon, being converted into pyruvate, a 3-carbon, interacting with coenzyme A to form acetyl-CoA, going into that citric acid cycle, creating lots and lots of NADH and FADH2, which go into the electron transport chain, pumping out lots of ATP through oxidative phosphorylation. This slide says it all. If you don't understand this slide, go back and learn the nitty-gritty subunits of it along the way. We've talked about all of these processes as in this lecture. So at this point, you should understand what's going on in here. And if not, then you need to go back and relearn it. Here are some reasons why glucose is the primary energy source in our cells. As we said, it's very small, it's quickly soluble, easily distributed through our body systems. It can provide quick ATP just from glycolysis itself, so oxygen does not have to be present, although only a small amount of oxygen is produced. Glycolysis is very important during peak levels of activity or exertion, especially in our red blood cells, or when tissue is deprived of oxygen. Glucose can actually be stored as glycogen, so it's nicely packed away in times of non-energy expenditure, and it can be easily mobilized by breaking down that glycogen through the process of, sorry, I heard a noise, through glycogenolysis. So you can pause the video here, go back and review this section. We're going to talk about lipids and how lipids compare to carbohydrates in our next little section. So I'll stop the video here, and we'll be back with lipids.